Good afternoon to one and all. The journey of 1,000 miles begins with one step. We are stepping towards a successful journey. On behalf of the Department of Life Sciences, Srimad Andavan Arts and Science College, Autonomous Trichy, we welcome you all for the second day of 2 days international webinar on current scenario in therapeutics, prospects and challenges. Hope yesterday's session was very informative and enlightened with the current advancement in the field of therapeutics. Let us start the second day session with the prayer.
वन्दामहे श्रीमदाण्डवन् पादुकाङ्कम् श्रीरङ्गक्षेत्रपवित्रोज्ज्वलपारिजातम् यत्र मानवाः सर्वे सन्मार्गदर्शिताः नित्यं पठन्ति ते विज्ञानकलाभाषाः पाठातिरिक्तविषयेऽपि प्रवृत्तास्ते सुशीलगुणावपि अनुचरन्ति नित्यम् नानाकलाक्रीडेष्वपि सुशोभिताः सर्वत्र सर्वदा ते आत्मप्रभावैः ओषधिवने स्थापित प्रयोगशालासु सम्यक् प्रकाशयन्ति ते स्वप्रभावम् अत्रैवाधीतयन्त्रविद्यासु निपुणास्ते विदेशेऽपि उज्ज्वलयन्ति शालाप्रभावम्.
It's my immense pleasure to invite the man of discipline and obedience, Dr. G. Sridharan, Associate Professor and Head, Department of Biochemistry, to welcome the second session. Welcome you, sir.
Warm good afternoon to all the participants. It is my great honor and privilege to welcome you all for the second day of the international webinar on current scenario in therapeutics, prospects and challenges, organized by the Faculty of Life Sciences, Srimad Andavan Arts and Science College. On this great occasion, I extend my heartfelt welcome to the speaker of today's first session, Dr. S. Sasikumar Purasamy, Postdoctoral Associate, Department of Oral Biology, School of Dental Medicine, University of Buffalo, New York, USA. We are very grateful for your presence, sir. I also welcome the second session speaker, Dr. R. Ramkumar Srivasadan, Research Associate, Department of Pharmacology, Case Western Reserve University, Cleveland, Ohio, USA. It is my immense pleasure to welcome our Honorable Secretary, Sri Amaki Balaji, the man of inspiration and positivity. I welcome you, sir. I would like to welcome all the delegates across the world for today's session. I hope everyone will find today's session helpful and interesting. Once again, I welcome you all. Thank you.
Thank you, sir. I cordially invite Dr. K. Atish, Assistant Professor in the Department of Biochemistry, to welcome and introduce our esteemed chief guest. Welcome you, sir.
A warm good afternoon to all the participants. I feel immense pleasure to introduce the guest speaker of this session, Dr. Sasikumar Purasamy. He pursued his bachelor and master degrees in biochemistry and later obtained his doctoral degree in the same discipline from Madurai Kamarajar University in the year 2015. After his PhD in 2016, he joined as postdoctoral associate in Department of Oral Biology at University of Buffalo, New York, USA, and continuing his position there till date. He was a meritorious student during his UG and PG days, and he excelled both in academics and research. His research instinct received him various research fellowships from UGC and DBT between the years 2009 and 2015, which molded his research career and granted him postdoctoral fellowship at University at Buffalo. His main area of postdoctoral research is investigation of microbial pathogenesis, particularly of oral pathogens, and photobiomodulation therapy to treat periodontal disease. Dr. Sasikumar published 16 research articles, 1 review article, and 3 book chapters in reputed journals and books. 4 more research articles are under review in high-index journals. To his credit, he also have a patent for multi-axle UVC device for optimal non-planar disinfection. He previously had academic membership with National Postdoctoral Association, American Association for the Advancement of Science, and American Society for Microbiology, and at present with American Society for Laser Medicine and Surgery to share its subject expertise. He is also acting as a reviewer in journals named Laser Surgery and Medicine, Applied Science, Biocatalysis, and Agricultural Biotechnology. Due to time constraints with this short intro, I hand over the session to our young and dynamic Sasikumar sir. Sir, now the session is yours.
So, thank you for your introduction, Dr. Atish. So this time I want to thank Dr. Radhika ma'am, Principal of Srimad Andavan and Arts and Science College. And I want to thank Dr. Jyoti ma'am, Dean of the Life Sciences, and HOD, Dr. Sridhar sir, Atish, and Dr. John sir, Dr. Siva Subramanian, and all other faculties for providing this great opportunity to share my one of research findings. So good afternoon to one and all. So I'm going to share my screen. So I hope everyone can see the slides. Hello? Hello?
Yes, sir.
So can you see my slides?
Yes, sir. Yes, sir.
Okay, thank you. Today I'm going to talk about one of my research findings. It's which entitled Molecular Rationale for Engineering Probiotics for Human Therapeutics. So today my talks divided into 4 main parts. The first part is background and objectives. Objective is subdivided into 4 different parts, and the conclusions and what are the ongoing projects. In the background session, we can briefly see so for what disease and why we engineering the probiotics. In the objectives, we can see what are the methodology we applied and what are the results we obtained from our research works. So in conclusion, we can see the output of our research findings and current projects. A couple of slides, we can see what are our ongoing projects and a list of publication and acknowledgements. So first we can start with the introduction. I hope everyone heard about the name kidney stone. It's a very common disease. It is affecting 6 to 12% of world population and 12% of the Indian population. There are mainly 3 different kinds of kidney stones. One is calcium oxalate, second one is calcium phosphate, and third one is uric acid stone. Among these 3 types of stone, calcium oxalate is a major stone. It's affecting 70 to 80% of the kidney stone population. So mostly the kidney stone population, so 70 to 80% population is experienced only the calcium oxalate. Only 20% of the population is experienced the other 2 different kind of stones. So what is the main risk factor for the formation of calcium oxalate is the amount of oxalate excreted in the urine. It's a very crucial risk factor. When the oxalate concentration is exceed 40 to 43 milligram per day in the urine, it is called hyperoxaluria. It is a major risk factor for the formation of the kidney stone. When the concentration is exceed 40 to 43 milligram per day, it will start to bind with the calcium and aggregate. The stone will deposit it in any part of the kidneys. So our stone will be formed. And other than this risk factor, our dietary contribution to urinary oxalate excretion is up to 50 to 60%. So there are 2 main sources for hyperoxaluria. One is our metabolic end product. Whatever we eating, the food is metabolized in the liver. The metabolic end product is oxalate. That is only 40%. Remaining 50 to 60% oxalate excreted in the urine is mainly derived from our food. Whatever we eating the daily basis, so based on that food, your oxalate concentration will increase and hyperoxaluric condition will form. there are a lot of foods containing a higher concentration of oxalate. For example, most of the green leaf and chocolate and the meat product have the high concentration of the oxalate. So what are the current treatment available for treating these hyperoxaluria and calcium oxalate is ureteroscopic stone removal. So in this method, they're using the endoscope techniques to break down the stone. The lithotripsy, this is by using the waves, using the ultrasonic lithotripsy and extracorporeal shock wave, they will break down the stones. The broken stone will be excreted in the urine. Other thing is they're using drugs to dissolve the stone. It's hydrochlorothiazide and sodium cellulose phosphate. These all are two common drugs used for the treatment of calcium oxalate. And some extreme case, they go for open surgery. And another case is dietary restriction. It means they have to eat low oxalate diet. But the condition is very difficult to eating the low oxalate diet because most of the food, we don't know what is the concentration of oxalate. The food also not labeled how much percentage oxalate is present in that. And also stop eating those kind of food also leads loss of other essential minerals and vitamins. So what is the disadvantage of existing treatment is by using the ureteroscopic and the lithotripsy, so it will cause the acute renal injury and a decrease in the renal function. And also it increase the risk for chronic kidney disease. And another one big disadvantage of this treatment is recurrence of the stone. More than 50% of the people experience their second stone within 5 years. Again, they have to go for a surgery or ureteroscopic stone removal. So repeated going will cause the stress in the kidney. So it will cause us kidney damage or chronic kidney diseases. And also some of the treatment is not up for those who are poor surgical patient. So best keeping this mind, so lack of effective treatments leads alternative treatment for calcium oxalate stones. Based on these, we designed a strategy to make the designer probiotics. So designer probiotics nothing but we designed a strategy to engineer non-oxalate degrading lactic acid bacteria into oxalate degrading lactic acid bacteria by simply cloning the special genes called oxalate decarboxylase from Bacillus subtilis origin. The function of oxalate decarboxylase is it will break down the oxalate into formate and carbon dioxide. So artificial colonization of these recombinant bacterium in the intestine, we can degrade the oxalate available in the intestine. So less oxalate will be absorbed and the hyperoxaluria condition will be stopped by lower excretion of oxalate by the kidney. So this is our hypothesis of the research. So how are we going to tackle this hyperoxaluria and the calcium oxalate stones? This schematic of the figure showing that so our oxalate is metabolized by the oxalate decarboxylate into carbon dioxide and formate. So mostly 70% of the oxalate absorbent is happening in the ileum and colon part of our intestines. So by colonizing our recombinant lactobacillus plantarum, which is degrading oxalate decarboxylase, we can metabolize, we can degrade the oxalate available in the intestine, so we can stop the oxalate absorbance from the intestine. So it leads to a very less oxalate excretion in the urine, so we can prevent the calcium oxalate stones. So this is our whole hypothesis of the work. So based on this, our work comprises 4 objectives. Objective 1 is to clone and secrete the heterologous oxalate decarboxylase in lactic acid bacteria. And objective 2 is to construct constitutively oxalate decarboxylase secreting lactobacillus plantarum and evaluate oxalate degradation in vitro. And objective 3 is to evaluate in vivo oxalate degradation efficiency of this recombinant bacterium in the rat model. And 4th objective is to develop biologically contained probiotics using Acknowledgement Grouptointron LLLKFB. So we can see one by one objectives. Objective 1 is molecular cloning and secretion of heterologous oxalate decarboxylase in lactobacillus plantarum for in vitro oxalate degradation. Excuse me. So in these objectives, first we choose an lactobacillus plantarum as a host strain for expressing our gene of interest. Why? Because the Lactobacillus plantarum is a human isolate. The bacterium is first isolated by Clearbezum et al. During the 2003 and the expression system availability. Because if you want to express one of your gene of interest into the host system L plantarum, you should have the tool to engineer the bacterium. So the expression system availability. So the expression system for Lactobacillus plantarum is well established by the Sorvik et al. 2003. So they already constructed a lot of plasmids and the cloning system expression pattern for the Lactobacillus plantarum, and the whole genome of this L plantarum was sequenced twice, and the most important is the signal peptide functionality was well studied. Why? Because we're going to express our gene of interest into the extracellular levels. To express the gene of interest into the extracellular level, you should know the optimal signal peptide, which is more prominent for secreting your gene of interest into the outside of your bacterium. And another important thing is this bacterium is resistant to physiological pH of the human intestines. So this figure shows the manipulation of the recombinant plasmid. So you can see these the PLB3050 amylase. This is a lactic acid bacterial expression vector. So PLB36 having our gene of interest oxalate decarboxylase. So we amplified this oxalate decarboxylase from this plasmid by using PCR method, and this was restriction digest with Sol1 and E-carbon. This restriction digest 1.2 kb our gene of interest was ligated into the plasmid, the lactic acid bacterial expression plasmid PLB3050, which also digest with EcoR1 Sol1, and it was ligated by using the T4 DNA ligase, and our resulting plasmid is called PLB3050OX36. So our gene of interest was just cloned downstream of the leader peptide PLB3050 and the induction promoter. So finally, this plasmid was transformed into the Lactobacillus plantarum, and we studied, after transformation, we studied the expression and secretion of this protein, and we studied the oxalate decarboxylase activity of this protein, and finally we studied the in vitro oxalate degradation efficiency. We can see one by one. So this figure A and B shows the construct we made in these studies. So you can see the PLB0373OX3C. So this PSPPA is an induction promoter. By using suppressing P induction peptide, we can induce this promoter. So once it's get induced, our leader peptide 0373 and our gene of interest will get expressed. We cloned our oxalate decarboxylase just downstream of this leader peptide. So these also the same way we constructed. The only difference between these two construct is carrying the leader peptide. So the figure A shows the leader peptide 0373. Another one is carrying the leader peptide 3050. So we're going to study which one is more efficient in secretion of our oxalate decarboxylase. So we studied in the STS page. So figure C shows the protein was resolved. The protein from the supernatant of wild type and our recombinant bacteria was resolved in the STS page. So the M is the ladder, the protein ladder, molecular weight in kilodaltons. One is the protein from the supernatant of the wild type bacterium, WCFS1. So as expected, there is no protein because the wild type bacterium don't have the oxalate decarboxylase. And 2 and 3 is a protein from the supernatant of our recombinant strain, which carrying the respective plasmid PLB0373OX3C and the PLB3050OX3C. You can see that 2 is from the recombinant bacterium carrying the plasmid PLB0373OX3C. 3 is carrying the recombinant bacterium carrying the plasmid PLB3050. As expected, we found that 44 kilodaltons size of that our protein of interest in the supernatant of the recombinant bacterium, but we couldn't see any protein equal to the size of OX3C. So this result suggests that our protein of interest was successfully actively secreted into the extracellular level by using the leader peptide 0373 and leader peptide 3050. Then we analyzed the oxalate decarboxylase activity by using different strain. So this table shows the activity. So you can see that this is a WCFS1 is a wild type lactobacillus plantarum. This acts as a controlled strain. So there is no oxalate decarboxylase present. So no, the protein was not produced. Therefore, no activity in culture supernatant and cell fraction and secretion efficiency was not determined. Whereas in the WCFS1 strain, which carrying the PZ-OX3C, this is our lab construct. So in this construct, our protein of interest was expressed into the intracellular level. So therefore, there is no activity in the culture supernatant, whereas the cell fraction showing 2.8 unit per milligram of protein. So secretion efficiency was not determined because it's not secreted into the outside. Interestingly, our recombinant bacterium carrying the PLB0373OX3C and the PLB3050OX3C plasmid expressing our protein of interest into the extracellular level. Therefore, we found the activity in the culture supernatant like 0.05 unit and 0.02 unit per milligram protein, whereas the cell fraction shows 0.18 and 0.21 unit per milligram of protein. Then we studied the secretion efficiency among these two recombinant bacteria. So we found that almost 22% of that protein expressed by the recombinant bacterium carrying the plasmid PLB0373 is secreted into the extracellular level, whereas other recombinant bacterium only secreted the 9% of the protein into the extracellular level. So these results concluded that the recombinant bacterium which carrying the PLB0373OX3C is more efficient in secreting and secreting our oxalate decarboxylase into the extracellular level. Then we studied the in vitro oxalate degradation capability of wild type and recombinant lactobacillus plantarum. Figure A shows the oxalate degradation efficiency of wild type strains. When the bacterium started to grow, the oxalate concentration in the culture supernatant was analyzed. In different time points, we analyzed the oxalate concentration in the culture supernatants. So we couldn't see any oxalate degradation in the wild type lactobacillus plantarum, whereas our recombinant bacterium carrying the plasmid, our construct PLB0373OX3C and PLB3050OX3C, showing the activity. When the cell started to grow, these arrows indicate the induction for expression of oxalate decarboxylase by using the induction peptide. When the cell started to grow, the oxalate concentration in the supernatant was gradually decreased in the different time points. So this clearly suggested that our recombinant bacterium carrying the plasmid PLB0373OX3C is almost degrading 5 millimole concentration of oxalate, whereas other recombinant strain only degrading 2.5 millimole concentration of oxalate. So in all our future studies, we plan to use this recombinant strain which carrying PLB0373OX3C. So objective 2. So it's constitutive expression of oxalate decarboxylase in L plantarum and in vitro oxalate degradation. So what's the difference between the objective 1 and objective 2 is, objective 1 is we are expressed our protein of interest by using the inducible expression. Objective 2 is aim is to constitutively express our oxalate decarboxylase in L plantarum, the in vitro oxalate degradation. Why this constitutive expression is needed is because in our next chapter, we're going to use this recombinant bacterium for in vivo studies. The inducible expression is only useful for only the table side, only you can use in the laboratory. If you want to study the function of your protein into the in vivo system, you have to have the constitutive expression system. That's why we plan to constitutively express our gene of interest. So for that, we just replaced the inducible promoter by using the constitutive promoter PLDHL. So to constitutively express, we isolated the genomic DNA from the lactobacillus plantarum, and the promoter of the lactate dehydrogenase gene was amplified by the PCR method, and the promoter PLDHL was cloned just upstream of the leader peptide. So the resulting plasmid is called PLDHL 0373 OXDC of 7.3 kb. So this plasmid was transformed into the lactobacillus plantarum, and we analyzed the expression pattern of our oxalate decarboxylase, and the activity also we studied. So figure B shows that the protein from the culture supernatant of different kind of bacterium. M is the protein ladder in kilodaltons. WCFS1 is protein from the culture supernatant from the wild type strains. So there is no protein, there is no oxalate decarboxylase genes. So there is no protein present in the WCFS1. Whereas PLDHL OXDC, this is our lab construct. This is expressing our gene of interest into the intracellular levels, not into the extracellular. So we couldn't see any protein of interest into the supernatant of that recombinant strain, whereas our construct carrying recombinant bacterium showed the 44 kilodalton of our oxalate decarboxylase in the supernatant. So this OXDC shows we purified the oxalate decarboxylase from culture supernatant of our recombinant strain. So both clearly showing that the expressed protein and purified protein is equal to 44 kilodalton of our gene of our protein of interest oxalate decarboxylase. The figure C shows the chromatogram generated during the purification of our oxalate decarboxylase by using the imidazole histidine tag methods. When the imidazole concentration is increased, the elution of our protein of interest was started between the fraction of 49 and 53. We pulled and we analyzed the oxalate decarboxylase activity. So similarly, we checked the oxalate decarboxylase activity in different strain, wild type strain and a recombinant strain. So WCFS1 is a wild type bacterium. So there is no protein, no activity present in culture supernatant and cell fraction. Whereas the recombinant strain, NC8OXDC, carrying our lab construct, PLDHL OXDC. So it's expressing our protein of interest into the intracellular level. So therefore, culture supernatant not showing any activity, whereas the cell fraction shows 0.27 units per milligram of proteins. Secretion efficiency was not determined. Whereas interestingly, our recombinant strain, WCFS1 OXDC, which carrying our recombinant plasmid, PLDHL 0373 OXDC. So it is successfully secreted our protein of interest into the extracellular level. So the culture supernatant showing 0.06 unit per milligram, whereas the cell fraction shows 0.18 unit per milligram. So similarly, the secretion efficiency also showing that the signal peptide 0373 is almost secreting 25% of our oxalate decarboxylase into extracellular level. So similarly, we analyzed the in vitro oxalate degrading capability of a wild type versus recombinant bacterium. When the cell started to grow, we couldn't see any oxalate degrading capability of wild type strain, whereas the recombinant bacterium is carrying our recombinant plasmid, showing the gradually decrease in the oxalate concentration to 120 hours. It almost degrading 12 millimole concentration of oxalate. So overall, this study suggested that our constitutive promoter is more efficient in expressing our oxalate decarboxylase, and our signal peptide or leader peptide 0373 is more efficient in secreting the oxalate decarboxylase into extracellular levels. So objective 3. Objective 3, in vivo oxalate degradation ability of recombinant L plantarum. So by using our already developed engineered recombinant bacterium, which constitutively expressing oxalate decarboxylase, we're going to check the in vivo oxalate degradation ability of this recombinant L. plantarum in the rat model. This figure shows the animal study designs. For this experiment, we choose a male Vistor albino rats weighing 130 to 140 grams. So rat was divided into 5 groups. Group 1 is a controlled rat, which fed with normal rat food. Group 2 to 5 rat groups received the rat feed, which mixed with 5% potassium oxalate. We commercially synthesized the rat food, which mixed with potassium oxalate. So group 2 rats only received the potassium oxalate diet. Group 3 rat is colonized with wild type L. plantarum from the second week onwards. Group 4 rat was colonized with our recombinant L. plantarum, which carrying the PLDHL 0373OX3C. So this is expressing our protein of interest into the extracellular level. Whereas the group 5 rats colonized with the recombinant plantarum, which carrying the PLDHL OX3C, so which expressing our protein of interest into the intracellular level. So every week, the day basal level, day 0, 7, 14, 21, and 28 days, up to 28 days, we collected the 24 hours urine sample and urinary biochemical parameter was analyzed. And the end of the experiment, rat was sacrificed and blood was collected and the serum parameter were analyzed and the kidney was dissected out and oxalate present in the kidney tissue was analyzed and the histopathology of the kidney tissue was analyzed with the help of a pathologist, and also the presence of calcium oxalate in the kidney tissue was visualized by using light microscope and polarized microscope. So this graph shows that urinary excretion. So urinary excretion of oxalate during different days of among the rat groups, different rat groups. In the day 0, so there is no difference between the rat group. Day 0 means it's basal level. So in the day 7 and 14, we can clearly see that rat received with the potassium oxalate diet is started to excrete more oxalates. So it excreting statistically significant of more oxalate when compared to control rat, group 2 to 5. We colonized the rat from the day 14. So by resulting of the colonization, you can clearly see that day 21 and 28 urinary parameter. So the calcium urinary oxalate excretion was gradually decreased in the rat group, which colonized with our recombinant bacterium, group 4 and 5. Whereas the non-colonized group 2 showing very high amount of urinary oxalate, and group 3 rats colonized with wild type bacterium also showing statistically significant higher amount of urinary oxalate. So overall, this result suggested that the rat colonized with our recombinant bacterium is excreting statistically lower oxalate when compared to non-recombinant bacterial colonized groups. Similarly, urinary calcium also observed in day 21-28. The urinary calcium was significantly reduced in group 4 and group 5 rats when compared to the non-colonized group versus wild type bacterium colonized group rats. We also analyzed the urinary urea. The end of the experiment, the day 28, so it's showing statistically significant the reduced urinary urea in group 4 and group 5 rats, whereas urinary urea was more in the group 2 and group 3 rats. Overall, this result suggested that the recombinant bacterium colonized groups showing statistically reduced urinary oxalate, calcium, and urea excretion. So this is crystal urea. So we visualized the presence of calcium oxalate crystal in the 24 hours urine sample. So the control rat doesn't receive any the food is normal food, so there is no calcium oxalate present. The group 2 rats received the 5% potassium oxalate, showing the presence of crystals. You can see the arrow mark, this showing the crystal of the calcium oxalate. In group 3 rats, which colonized with the wild type bacterium, also showing the presence of calcium oxalate crystal, whereas the group 4 rats received our recombinant bacterium, which is secreting oxalate decarboxylase into extracellular, showing the absence of calcium oxalate crystal, whereas in group 5 rats, which received the intracellular OXDC expressing bacterium, also showing presence of very less number of calcium oxalate crystal in the 24 hours of urine. End of the experiment, we also quantified the presence of kidney oxalate. When compared to control rat, the kidney oxalate was higher, statistically higher in the group 2 and the group 3 rats, but the recombinant bacterium colonized group shows statistically reduced level of oxalate in the kidney tissue. This is the visualization of a calcium oxalate crystal by using a polarized microscope. So the control rat, so there it's not received any potassium oxalate diet, so it's a healthy rat, so there is no calcium oxalate present. So in potassium oxalate group, this rat received we can see in the figure B. So in the black color dot is a pessalato staining of the calcium oxalate. It's confirming the presence of calcium oxalate, and also you can see the white color dots. These all are a presence of calcium oxalate. Whereas in the group 3 rats, which received the wild type Alplantarum, also showing the presence of the calcium oxalate, also showing the presence of calcium oxalate in the block colors you can see, and the presence of dotted small white color dotted also presence of calcium oxalate crystal. Whereas our the group 4 rats, which received the recombinant bacterium expressing our gene of interest in extracellular, showing the absence of the calcium oxalate, whereas the group 5 rats, which received the colonized with the recombinant bacterium expressing oxalate decarboxylase intracellular, also showing the very few crystals present in the kidney tissues. Overall, these studies, in vivo studies, suggested that colonization of the rat with our recombinant bacterium, which is secreting oxalate decarboxylase extracellular, is more efficient in reducing the urinary oxalate excretion, and also it stopped the formation of calcium oxalate crystal in the kidney tissues. So objective 4 is development of biologically contained probiotics. So what's the biologically contained probiotics, you can ask. So the final goal of any designed probiotic is obviously to use in the human or animal to counteract or prevent the disease. But the use of this genetically modified organism in the medicine raises the legitimate concern about the survival and propagation in the environment and the presence of antibiotic selection marker and other genetic modification to other organisms. So we have to stop the survival and propagation of these bacterium into the environment. So for that, the development of biologically contained, genetically modified lactic acid bacteria of inexpensive, easily administered therapeutic with a minimal side effect is highly desirable. So based on this, we plan to develop a biologically contained bacterium. So this bacterium only will survive into the human intestine. Once it released into the environment feces, it should not survive. For that, we designed a strategy to mutate the thymidylate synthase gene. So by mutating the thymidylate synthase genes, you can avoid the propagation of this bacterium into the environment. So in the environment, the absence of thymidine, it will not proliferate, it will not grow outside in the human body. It will only grow and colonize only in the intestine of the human. Based on that, we applied the tool called Grouptointron expression cassette to mutate the gene thymidylate synthase of the Lactobacillus plantarum. So we constructed the novel vector called the PLB-ACD4C. So in this vector, this is a lactic acid bacterial expression vector. So in this vector, we cloned the intron LLLTRB and the gene LTRA. This is a mobile genetic element. So by modifying these intron base pairs between the HIN3 and BS or G1, you can retarget this intron in any region of your gene of interest in the genome of Lactobacillus plantrum. So we plan to target the thymidylate A synthase gene in the genome of Lactobacillus plantrum. So we modified. So you can see the modified region in the green colors. So we retargeted these mobile elements. So what's get expressed by using inducible expression, these mobile elements will go and scan. So it will find the thymidylate A synthase. So it will mutate and it will insert into the thymidylate A synthase. So this figure shows the retargetron integration of thymidylate A synthase. This A is a wild type LLLTRB. It's a group 2 introns. Here you can see that this is a wild type base pairs, exon binding site 2 and exon binding site 1, intron binding site 2, intron binding site 1. This is a wild type base pairs. By modifying this base pair, you can target this intron into thymidylate A synthase gene in the genome of the Lactobacillus plantrum. So by using the computer algorithm, we modified the exon binding site 1 and the exon binding site 2 from this base pair to these base pairs. Then this region was cloned and transformed into the Lactobacillus plantrum and expressed by inducible expression. After inducible expression, these mobile elements get expressed and it will read the genome, and it will when it finds the Thia gene, it will start to integrate between the 188 and 189 base pairs of the thymidylate synthase. So you can see the figure C, the resulting construct cassettes will be, your intron will be integrated. So your gene of interest will be get mutated. So we created the Thia mutant based on by applying this Grouptointron technology. So then we analyzed the in vitro viability of this Thia mutant Lactobacillus plantrum. So in the absence of thymidine, so the growth of the mutant strain was depleted. When we increased the concentration of the thymidine, the growth also gradually increased in compared to the control wild type Lactobacillus plantrum. So the figure 12 shows the schematic representation of wild type and mutant Thia locus. So we modified the wild type Thia locus from 951 base pairs into 1.9 kb base pairs. So it will be get integrated between the 188 and 189 base pairs. So your Thia gene was mutated and the protein of interest, thymidylate synthase, was truncated. So there is no protein. So we can control the survival of this bacterium. So based on this, we can colonize this bacterium into the intestine. So it only survive into the intestine. When it is released into the environment, it will not survive because of absence of the thymidines. So the overall conclusion of this research is indicate that the daily oral administration of OXDC, secretory L-plantarum in rat, associated with the decreased excretion of urinary oxalate and reduced the risk of calcium oxalate crystal formation by degrading intestinal oxalate and leaving less oxalate available for absorbance. So in couple of slides, we can see the briefly we can see the what is the ongoing current projects. So in one of my research, we currently studying the bacterial pathogenesis of the oral pathogen called Tenarella forsythia. This is a oral pathogen. So it will cause the periodontal disease in our oral cavity. So the beauty of this bacterium is this bacterium don't have the peptidoglycan synthesis pathway. So if you try to grow this bacterium alone, it will not grow. It will only grow with some other bacteria. So how it's surviving into our mouth is in the presence of the fusobacterium. So when the bacteriums died, it will release a lot of peptidoglycan in the surrounding areas. This Tenarella forsythia, this bacterium Tenarella forsythia, secreting the special enzyme in the group of hydrolase, it will secrete it into the outside. It will degrade the available peptidoglycan in the outside by using that enzyme. They updating those peptidoglycan from the surrounding. Then they're using those subunit for making the peptidoglycan synthesis pathway in the Tenarella forsythia. So we plan to target those novel genes. So by mutating those novel genes, we can stop the growth of this Tenarella forsythia. For that, we cloned and expressed and purified the hydrolux family gene BF047 from the Tenarella forsythia. So our purified BF0747 showed peptidoglycan degrading activity by zymography. So this purified protein is responsible for degrading the peptidoglycan available in the outside. So by using these enzymes, this bacteria is recycling the peptidoglycan. So by making the inhibitor, for targeting the inhibitor for these genes, we can develop the therapeutics for the periodontal disease. This is an ongoing project. So this is a preliminary results we completed. So another research we are working is a photobiomodulation therapy. This is a very emerging therapeutic use for regenerative medicine. So photobiomodulation therapy is nothing but by using the light as a therapeutic tool, you can induce the regeneration mechanism. So already in our lab, by using photobiomodulation therapy, the wound healing and oral mucositis. Hello? Hello? So sorry for the interruption. So photobiomodulation therapy. So what is a photobiomodulation therapy? It's nothing but using a light as a therapeutic cues. Light as a therapeutic cues for inducing the regenerative medicines. Already in our laboratory, by using low-level lasers, we showed the wound healing and oral mucositis. So currently we are optimizing this photobiomodulation therapy, otherwise called low-level laser therapy, to treat oral and burn wound and oral mucositis. Previously in our laboratory, also we found the expression of ATF4 gene. When the tissue is exposed to the laser light, it will start to express the ATF4 protein. So it's act as a phototoxicity biomarker. So in these studies, one of our studies, we studied the signaling pathway by which this ATF4 is expressing in epithelial cell, fibroblast, macrophage, and neuronal cell lines using immunoblotting and immunostaining methods. Our preliminary results showed that the inhibition of TGF-beta1 pathway via the Srimad, NF-kappa-B, and ERK and P38 pathway is a key to laser-mediated ATF4 expression. So in another study, interesting studies, we collaborated with the company called Mureva. So we developed a device called the OroGlow LED device. So for the treatment of oral mucositis. So oral mucositis, I hope you know what is oral mucositis. It's nothing but when the oral cancer patients treated with radiation therapy and a high amount of chemotherapy, the oral mucosa get damaged and the fibrosis will form, the scar tissue also formed. Because of this scar tissue and fibrosis, they cannot taste the food and also they cannot open the mouth. So the quality of that patient is decreased. To cure these oral mucositis, we developed a device called OroGlow LED device. So it will emit the particular wavelength of the light in the particular dose level, optimal dose levels. So it will induce the regenerative by using the neighbor stem cells. So your scarred tissue will get regenerated. So based on these things, we studied the proliferative efficacy of normal oral keratinocytes versus the cancer cells, oral squamous carcinoma cell by in vitro and in vivo model using a newly developed device, OroGlow LED light. You can see the device. So the device is already ongoing the clinical true trials. So our preliminary results show that low power light induced the cell proliferation in normal keratinocytes, but it's not inducing the cell proliferation in the cancer cell lines. So we studied the efficiency of this OroGlow light in the nude mice, in vivo nude mice studies. So we almost we bought almost 100 nude mice. Each mice is cost almost $200. We almost spent $20,000 for buying these nude mice. You know, the nude mice is very sensitive to that environment and pollutant. So this mice is maintained in the very sterile room. So this mice is breathing only the filtered air. So from the HEPA filtered air, and they receive the very sterile and filtered water, very hygienic food, and you have to cover everything like treating COVID patient. You have to cover your hair and your nose, mouth, and everything, your food, everything only is allowed to, your eyes only allowed, you can use your eyes. So this mice is maintained in the very sterile condition, hygienic conditions. So we injected the oral cancer, by injected that cells, oral squamous cancer cells in that mice, and we treated the mice, you can see in the figures, we treated the mice with this light almost for different groups received the different dose of the light, and we analyzed the growth of the cancers in those mice in the end of that experiment. So our preliminary study shows that so the growth of that cancer is very low when the mice is treated with light when compared to non-treated mice. So still the studies are ongoing and clinical trial also ongoing. So I couldn't share a lot of results from these studies. So in another project, we studied the disinfectant efficacy of UVC device. We developed a novel UVC device to disinfect the non-planar surface. So only treating the planar surface means even though it is a 3D structure, it will treat all the parts by applying this UVC device. So we studied the preliminary result was obtained and we filed the patents. This device was developed with the help of Applied Research System Canada. So these all are some of my publication associated with my projects. So acknowledgement. So I want to thank Professor G.S. Elmum for his guidance. And I want to thank Dr. Guy, Michael, Stephen, and Alan for providing different construct and different plasmids. Also, I want to thank my boss, Arani. You can see he's Arani, Praveen Arani, for introducing me into the different field of biomodulation and regenerative therapies. So you can see this me, and these all others are a graduate student of our research groups. So thank you. Hello.
Thank you, sir. Thank you for your valuable speech. Students
Thank
have
you.
raised a few questions. The first one is, what is the role of the leader peptide?
So the role of a leader is nothing but it will guide your protein of interest into the extracellular level. So it's a signal peptide. So mostly protein is expressed, the bacteria is expressing protein in two ways. One is intracellular, within the cells. The function of leader peptide is it will drive your protein of interest into the outside of the cells. It will excrete. It will be present in the supernatant, not in the inside of the cells. That's the role. Why? Because we want to degrade oxalate into the extracellular levels. When you eat the food, the dietary oxalate will be available. So we want to degrade that extracellular oxalate. That's why we plan to secrete the protein of interest into the extracellular level. I hope they got it.
Yes, sir. The second question is, what is the difference between inducible expression and constitutive expression?
Yeah. The main difference for inducible expression is inducible expression is like IPTG induction. You have to give externally some peptide, very 20 amino acids peptide like that. By synthesizing that peptide also very expensive. So you have to give that peptide. That peptide will bind to that inducible promoter. Then only your gene of interest will get expressed. That is called inducible expression. What constitutive expression is, without using any signal peptide, your gene of interest will automatically get expressed continuously. That is kind of continuous expression of your gene of interest. Because why? Because if you colonize your bacterium into the rat model, you cannot give the peptide for inducible peptide. It also very expensive. We don't know it will go and reach that. That's why we developed the constitutive expression system. So we don't need to give any peptide. So it will, your gene of interest continues automatically get expressed in intestine. That's why we changed from inducible expression to constitutive expression.
Yes, sir. The final question is, what is the role of probiotics in human health? Because nowadays probiotic curd also commercially available. So tell me some information about role of the probiotics for human health.
The role of probiotics means we can tell that without probiotics also we cannot survive. So they play that kind of major roles in our maintaining our body health. So the bacterium present called microbiome or microbiota or whatever the bacterium present in our body, the total number of bacterium present in our body is more than 100 trillion cells. This is the number is more than our total number of cells in the body. They're playing a lot of roles. For example, if eating some of the protein or some carbohydrate, lot of these things is degraded by your probiotics and some other, even we don't know, a lot of other bacteria still not explode. Still the millions of bacteria in the intestine, they're degrading. Then only your gut will get absorbed. Without degrading, without help of this probiotic, your gut will not absorb. They will not take those nutrition. So it's playing a major role in your immune system also. Your immune system also built based on what are the bacterium you have in your intestines. So your cancer also based on the number of probiotics is less, other pathogen is high, so it will lead to the cancer. Even nowadays, a lot of research showing the absence of particular probiotics leads to diabetes, brain loss of function, brain tumor, cancer, etc. So without probiotics, we cannot live. So that much role probiotics play in our health and disease. It's preventing a lot of diseases. If the pathogen is entered, so it will stop the proliferating of those pathogens. So even COVID also, we don't know, maybe our probiotics play a role. That's why somebody is having the more resistant than other people. So in future, you will see a lot of articles and findings.
Sir, one more question. What is the difference between the probiotic and antibiotics?
Yeah, this is totally different. Probiotic is a bacterium. It's a living molecule, living strain. Antibiotic is a chemical compound. So antibiotic for some fever to kill the bacterium. So antibiotics is to kill the bacteria. That is called antibiotic. That's why it's called antibiotic. It will act against the bacterium. Probiotic is a bacteria.
Thank you very much, sir, for your valuable speech. And on behalf of the organizers, we once again thank you all. Thank you very much for the excellent and informative scintillation speech. Thank you, sir.
Thank you very much. Thanks for Dr. Sridhar and Radhika ma'am, Jyoti ma'am, and all other faculties for this great opportunity. Thank you.
Thank you, sir. Dear participants, you are all join the fourth session exactly at 4 p.m. Thank you.
A pleasant evening to one and all. I welcome you all for the final session of the two-day international webinar on current scenario in therapeutics, prospects and challenges. Now I invite Ms. G. Vanitha Mani to introduce the speaker of this session.
A pleasant good evening to everyone. I welcome you all to the second session of the webinar on current scenarios in therapeutics, prospects and challenges. It's a great honor for me to introduce the speaker of this session, Dr. Ramkumar Srinivasan, Research Associate, Department of Pharmacology, Case Western Reserve University, Cleveland, Ohio, USA. He started his college studies in 2007 in the field of biotechnology, completed his bachelor's degree in J.J. College of Arts and Science, Pudukkottai, Bharathasan University, and his master's degree in K. Rangasamy College of Technology, Anna University, Tiruchirappalli, Tamil Nadu. Ultimately, in 2014, he achieved highly commended PhD in the field of microbial biotechnology in Bharathasan University. He has vast research experience. He acted as a postdoctoral scholar from 2015 to 17 in the Department of Pathology and postdoctoral fellow from 2017 to 19 in the Department of Pharmacology in Case Western Reserve University, Cleveland, Ohio, USA. He has published 20 research papers, including one book chapter with total impact factor of 40.1. Professionally, he worked as a member in ARVO, Association for Research in Vision and Ophthalmology, from 2015 to 17. Again, he resumed as a member in ARVO from 2020. He is the person with the numerous awards and achievements. During his doctoral studies from 2010 to 14, he worked as a technical person in many hands-on training in advanced molecular biological methods and techniques in Bharathasan University. In 2012, he was awarded as DST Young Scientist Project Fellow by the Department of Marine Biotechnology, Bharathasan University. During the same period, he have attended the 37th annual meet of the Society for the Study of Origin and Evolution of Life, Japan at Osaka University of Pharmaceutical Science, Osaka, Japan. Also, he attended 51st annual meet of the Japanese Society of Ophthalmological Optics in Tokyo, Japan. He was awarded as a Indo-Japan Society for the Promotion of Science Project Fellow in 2012. He received numerous research articles in many prestigious journals of publishers like Springer, Elsewhere, Bentham Science, and MDPA. In 2017 and 18, Dr. Ramkumar Srinivasan awarded as Outstanding Reviewer in 2 international journals of Elsewhere publisher. Currently, he is a reviewer board member in Nutrients Journal, MDBA publisher. In 2018, he was awarded a travel guide to attend Carotenoid Golden Research Conference in Nadu, USA. In 2019, he was invited as a keynote speaker in an international conference and delivered many invited talks in well-reputed colleges in India. Also, he acted as a poster judge in symposium and a research showcase in Case Western Reserve University, Ohio, USA in 2019. Today, we are privileged to have an opportunity to listen to Dr. Ramkumar Srinivasan this evening. On behalf of Life Science Departments, Srimad Andavan Arts and Science College, Trichy, I kindly request you, sir, to initiate your presentation. Please, sir.
Thank you, ma'am.
Session is yours. Thank you.
Yeah, thank you, ma'am. Thank you for the nice introduction. Good evening to everyone. It is a very refreshing morning for me because it is here at 6 o'clock. So this is my first ever talk in the morning at 6 o'clock. Okay, let's get into the talk. Okay, I'm going to present today how the vitamin A homeostasis was regulated in the intestinal barrier. At first, I would like to give background information regarding vitamin A. Vitamin A is a lipid-soluble vitamin which we can get through the carotenoid in our diet. These carotenoids are 40 carbon chain isoperonide compounds. Most are lipids, which can act as provitamin A molecule. So for carotenoid to act as a provitamin A molecule, it must possess at least one single molecule of beta-inon ring. So some of the other carotenoids which has a modified beta-inon ring can give rise to the wide range variety of carotenoids. Out of these many carotenoids, Alpha carotene, beta carotene, and beta cryptoxanthin. These three can act as a provitamin A molecule because it has at least one beta ion ring. Out of these three carotenoids, beta carotene is the major and widely present carotenoid in our diet. And further, the other carotenoid which has a modified ring which is unable to act as non-provitamin A carotenoid, but it can act as a xanthophyll, especially lutein and zeaxanthin. These two carotenoid, which is highly present in our body in the retina region of our eye, and it protects retina from blue light induced retinal damage. So, as I mentioned earlier, beta carotene is the highly available carotenoid present in our diet. It was metabolized in our body into three different forms of vitamin A. The three different forms of vitamin A are all-trans-retinoic acid, all-trans-retinal esters, and 11-cis-retinol. These three different forms of vitamin A has a different physiological function. Let's see what are the different physiological function of these different forms of vitamin A. So when we take beta carotene as a dietary source, this was observed in the intestine and was first cleaved into two molecule of retinal by the enzyme beta carotenase 1. So this all-trans retinal was further converted to all-trans retinol, which is the circulatory form of vitamin A. And this all-trans retinol were further oxidized to all-trans retinoic acid. This all-trans retinoic acid is the active form of vitamin A, which is present very minute level up to only picomole level in our body. This active form of vitamin A binds to the retinoic acid receptor in the nucleus, and which controls up to 500 gene regulation. Most of the genes involved in carotenoid differentiation, embryogenesis, and reproduction, spermatogenesis, and immune cell maturation. Next, this all-trans retinol was further converted into all-trans retinyl ester by the enzyme LRAT. This LRAT is the main key player of the current talk, and I will present the importance of LRAT in the future slides. So this all-trans retinyl ester is the transport form of vitamin A, and also it is the storage form of vitamin. This all-trans retinal esters also act as a precursor for the formation of 11-cis-retinol. This all-trans retinol is storage form of vitamin A, which stores in the liver, especially in the stellate cells, and also in the fat. As this vitamin A was stored in the liver, when the lacking of all-trans retinal esters leads to vitamin deficiency, and also it leads to liver-induced metabolic diseases. And next, the very important form of vitamin A is 11-cis-retinol. This 11-cis-retinol, which is present in a very high concentration, about millimolar concentration, it's present only in the retinal region of our eye, because the retina of our eye has photoreceptors. One is rods and cones. These rods are important for our night vision. Cones are for color and morning vision. So this 11-cis-retinol binds to the opsin protein moiety of rhodopsin. When the light hit, this 11-cis-retinol were isomerized to all-trans retinol and generated nerve signals. This process is called phototransduction. This phototransduction induces a nerve signal, and it passes through the brain. Thereby, we are enjoying vision during our night. So now you come to know the importance of vitamin A. Hence, vitamin A is a micromolecule which is important for our normal physiological function. The lacking of vitamin A leads to vitamin deficiency and different disease conditions. According to World Health Organization, vitamin A deficiency is still a major problem in lot of underdeveloped and poor countries. If you see this map, India is still has a under vitamin A deficiency. So personally, I believe as an Indian researcher, it is important for us to know the how vitamin A was absorbed and metabolized in our body. For this reason, I selected this topic to present for the today's talk. Okay, now you know the importance of vitamin A. I have a question here. How does this vitamin A get into our body? Because vitamin A was unable to produce in our body by its own. So we always rely on the dietary source for the vitamin A. There are two forms of vitamin A. One is preformed vitamin A, which is from retinal ester, which is from animal source. Another one is provitamin A carotenoid, which is from the vegetable source. And this are the provitamin A carotenoid is the major form of vitamin A in the poor and underdeveloped country. The reason is to attain daily intake value of vitamin A, you have to take at least 25 gram of liver or half gallon of milk or 200 pounds of beef patty every day in the retinal ester form. Do you think it is possible? I don't think so. But in the meantime, other hand, if you see here, to attain the daily value of daily intake value of vitamin A, you have to take at least 100 gram of sweet potato or like carrot or some things. I think this is possible. That's the reason provitamin A carotenoid is the major form of vitamin A throughout the world. And hence, we are completely dependent on vitamin A. Hence, we are completely dependent on a dietary source for the vitamin A. The key step is to proper absorption and distribution of vitamin A in the intestine. The proper absorption and distribution of vitamin A involve multiple steps. It starts with the intestine, where the beta carotene were absorbed and converted into two molecules of retinol by the enzyme beta carotene oxygenase. And this retinol molecule were esterified by the enzyme LRAP and were converted into retinal ester, a transport form of vitamin A, and it's circulated in our body. And these retinal esters were further stored in the liver. And in the absence of dietary vitamin A, this liver produces this releases the stored vitamin A as a retinol molecule, and this retinol molecule bonds to the retinol binding protein 4, and this retinol binding protein 4 delivers the vitamin A molecule to the peripheral tissue, like the ocular region. So it is important to regulate the vitamin A in our body, because vitamin A is vital for our vision and then immunity and normal physiological function. There are a lot of research that focused only on how vitamin A was metabolized and homeostasis in the ocular region. Even in our lab, our main focus is how vitamin A's get into the eye. And as a second aim, our lab, we are focusing now how this vitamin A was regulated in the intestine. So throughout this talk, I'm going to genetically dissect how vitamin A was absorbed and metabolized in the intestine. Before that, it is important to know how this provitamin A carotenoid were absorbed and converted into vitamin A in the intestine. So when in the normal physiological function, beta carotene was absorbed into the intestine by the transmembrane protein SRB1, and this single molecule of beta carotene was converted into two molecules of retinaldehyde by the enzyme beta carotene oxygenase 1. A major retinaldehyde molecule were converted into retinal ester, the transport form of vitamin A, by the enzyme LRAP, and very minute by the enzyme Retargetron. after the retinal esters were formed, this was further packed into the chylomicron for the bodily distribution and liver storage. In other hand, this beta-carotene was converted into retinaldehyde and further oxidized to retinoic acid. Retinoic acid is a active form of vitamin A, and this active form of vitamin A is important for maintaining up to like 500 genes, which is important for regulation of 500 genes. It is important to maintain these retinoid homeostasis in the intestine, because especially retinoic acid, because retinoic acid is a double-edged sword. Appropriate level of retinoic acid in the intestine is important for the gut immunity and cellular differentiation in the intestinal epithelium, and this retinoic acid can act as an anti-inflammatory agent in most of the gut diseases. In the other hand, very low or a very high level of retinoic acid lead to hyper and hypovitaminosis, impaired gut immunity, oxidative stress, and impaired of intestinal epithelial integrity. So it is important to maintain appropriate level of retinoic acid in our intestinal barrier. And it is already known, just now I explained that retinoic acid is important for maintain the expression of 500 genes. In the intestine, this retinoic acid regulates one of the important genes that controls the beta-carotene absorption and conversion into vitamin A. The name of the gene is ISX. This ISX is a retinoic acid gatekeeper that controls the beta-carotene absorption and conversion into vitamin A. This ISX is a 241 amino acid homeodomain transcription factor, which is highly expressed in the intestine. When the beta-carotene was converted into retinaldehyde, this retinaldehyde was further oxidized to retinoic acid. This retinoic acid travels to the nucleus and bonds to the retinoic acid receptor, and that activates the transcription expression of ISX. This ISX was first discovered in Ramesh Sivadarsani lab in Harvard, but recently in our lab we discovered this ISX bounds to the promoter region of SCAR1 and BCO1 gene. Further, we discovered another thing, like this ISX can act as a repressor for the BCO1 in the in vitro promoter reporter assay. And in the in vivo, when the ISX were knocked out, there was a complete upregulation of SCAR1 and BCO1. So our data clearly shows that ISX is the negative regulator of SCAR1 and BCO1. So this negative feedback regulation guarantees the absorption of beta-carotene and conversion of beta-carotene into vitamin A in the intestine. To further confirm this mechanism, in our lab, we have generated ISX knockout mice, and this ISX knockout mice and wild-type mice were fed with beta-carotene, and we found there is up to like 10 to 30-fold increase in the accumulation of vitamin A in the ISX knockout mice fed with beta-carotene in the different region of intestine. And this accumulation of vitamin A is not only restricted in the intestinal part, but also it mirrored in the other part of tissue like liver, lung, and white adipose tissue. So when you see here, when the ISX knockout mice fed with beta-carotene has a very high level of vitamin A accumulation. Even this is not in the ISX knockout mice fed with the preformed vitamin A. This data clearly shows that the high accumulation of vitamin A in the ISX knockout mice is due to the conversion of beta-carotene into vitamin A. This data clearly shows that ISX act as a negative regulator for BCO1, a vitamin-forming enzyme. Overall, our study clearly shows that ISX is the key regulator for the regulation of vitamin A in the intestine. this ISX knocked out leads to the uncontrolled production of vitamin A in the intestine. On the other hand, when there is a premature upregulation of ISX, that leads to uh, complete downregulation of vitamin A forming enzyme BCO1. Finally, that leads to vitamin A deficiency. So, this data clearly shows that it is important to maintain maintain the ISX expression in the intestine for the proper vitamin A metabolism and homeostasis in the intestine. So, now we know the ISX is most important for the proper vitamin A homeostasis in the intestine. What regulates the ISX? If you see this pathway, the retinoic acid is the one which regulates the ISX expression. Okay, here there is one more question which need to be answered. How does this retinoic acid concentration regulated in the intestine? The normal, when you look at this pathway, the normal physiological process, and the beta carotene was converted into two molecules of retinaldehyde by the enzyme beta carotene oxygenase 1. Majority of the retinaldehyde were converted into retinal esters for the bodily distribution. By this way, this retinoic acid were kept in appropriate level. This conversion of retinal molecule to retinal esters is mediated by the enzyme LRAT and minimal by the enzyme DGAT1. So, based on this, we hypothesize that LRAT is the enzyme which indirectly regulates retinoic acid level in the intestine. Okay, before entering into the talk, it is my privilege to introduce about the history of LRAT, because this LRAT has a very long history with our department. This LRAT enzyme was discovered by Professor Paul McDonald, and the enzyme structure was determined by Martin Kolchak. These two guys were in our which they are worked in our department, and we are the long-term collaborator. Still, we are working with them. Thanks for them for this introduction. That's how I can able to present my talk here. Okay, this LRAT is dominant acyl transferase, which catalyze the formation of retinal ester by transferring the acyl group of SN1 position of phosphatidylcholine to the retinol molecule. That retinol were converted into retinal ester. This is how this LRAT works. So now get into the talk. So to see how this LRAT is important for beta carotene metabolism, for this experiment, we initially carried out a very short-term experiment. For that one, we use wild type and the LRAT knockout mice. After wean, these mice were fed with vitamin A deficient diet for 1 week. The reason for using vitamin A deficient for 1 week is to complete deprivation of dietary vitamin A in the intestine. After this washout period, these mice were randomized into two groups, where one group were fed with vitamin A sufficient diet and another with beta carotene diet. After 3 days of intervention, these mice were sacrificed and their tissue were used for measure the vitamin A level and gene expression level by qPCR. So for the vitamin A level measurement, we use a HPLC system. So the peak 1 and peak 2 represent the retinal ester and retinol respectively, and this were confirmed by the respective spectral properties. And peak 3 represent the beta carotene, and this also confirmed by the spectral properties. Initially, we measured the retinol levels in the intestine of the wild type mice. The wild type mice fed with vitamin A sufficient and beta carotene has a similar level of vitamin A, which confirms that both preformed and provitamin A can able to observe similar level in the wild type mice. In contrast, this LRAT knockout mice fed with vitamin A sufficient diet has very low level of vitamin A compared compared to the wild type, even worse in the LDAT knockout mice fed with beta carotene. Next, we determine the retinal ester level in the intestine. Hence, this wild type mice has LRAT in their body. The dietary vitamin A can able to convert it into retinal esters. As expected, there is a very low level of retinal ester present in the LRAT knockout mice fed with vitamin A sufficient. There is no retinal esters present in the beta carotene fed LRAT knockout mice. You may ask the question, hence this LRAT knockout mice don't have LRAT, how come this there is accumulation of little retinal esters present in the LRAT knockout mice fed with vitamin A sufficient? The reason is, as I already explained here, you can see here, this is the enzyme DGAT1, which can able to act as alternative for LRAT, but in very less extent. That's why you can see here a very less amount of retinal ester in the LRAT knockout mice fed with vitamin A diet. So this data shows that there was a problem in the LRAT knockout mice fed with beta carotene, and it has a problem in the beta carotene metabolism in the LRAT knockout mice. To confirm further, we measured the beta carotene level of the intestine of wild type mice and LRAT knockout mice fed with beta carotene, and we found there is accumulation of beta carotene in both wild type and LRAT knockout mice in the less extent. So next, we would like to measure the vitamin A level in the liver, because liver is the major vitamin A metabolism tissue. Compared to the wild type mice, this LRAT knockout mice fed with vitamin A sufficient and beta carotene has a very less amount of vitamin A. In contrast, this LRAT knockout mice fed with beta carotene has a high accumulation of beta carotene. This data clearly shows that this beta carotene was absorbed intactly in the intestine and the liver of LRAT knockout mice. Next, I would like to measure the gene expression of the retinoid metabolism gene in the intestine. As I already mentioned, ISX is the key regulator of vitamin A homeostasis in the intestine. We found here there is a huge upregulation of ISX in both LRAT knockout mice fed with vitamin sufficient and beta carotene compared to the wild type. This data clearly shows there was a profound effect between genotype between wild type and LRAT knockout in the ISX expression. As I already mentioned, ISX is the negative regulator of vitamin A forming enzyme BCO1. As we expected, this LRAT knockout mice has a complete downregulation of vitamin A forming enzyme BCO1 expression. This short-term experiment clearly shows that LRAT knockout mice can able to absorb beta carotene intact. Due to the downregulation of BCO1, a vitamin forming enzyme, it is unable to convert into vitamin A. But it is very difficult to come to a conclusion with this short-term experiment. We would like to know why this LRAT knockout mice are unable to convert beta carotene into vitamin A. For this experiment, we carried out a long-term experiment. For this, we used only LRAT knockout mice. In the long-term experiment, we take LRAT knockout mice. After these mice were fed with vitamin A deficient for 1 week. Afterwards, these mice were randomized into 3 groups. One group were fed with vitamin A sufficient diet and another with beta carotene diet. And the last group were continuing with vitamin A deficient diet. This group will act as a basal control. After 4 weeks of intervention, these mice were sacrificed for analyzing vitamin A level and gene expression level for the vitamin A metabolism. So when we measure the retinol levels of the intestine, we found that there is a high level of vitamin A in the vitamin A sufficient LRAT knockout mice. in both retinol and retinal esters. And we found there is a minimal level of vitamin A in the beta carotene supplemented LRAT knockout mice, but there is undetectable level of excuse me. There is an undetectable level of retinal esters in the beta carotene supplemented mice. In contrast, there was a high accumulation of beta carotene in the LRAT knockout mice fed with beta carotene. So, as expected, there is no retinol, retinal esters, and beta carotene in the vitamin A deficient eye. So, this data clearly shows that beta carotene was able to accumulate intact in the intestine of LRAT knockout mice. So, to know the molecular mechanism why this beta carotene were unable to convert into vitamin A in the intestine of LRAT knockout mice, we would like to check the genes involved in vitamin A metabolism in the intestine and their expression by qPCR analysis. So, as I already mentioned, ISX is the key regulator for vitamin A metabolism in the intestine, and we found that there is a complete upregulation of vitamin sufficient, complete upregulation of ISX in the vitamin sufficient and beta carotene diet. Hence, this ISX is the negative regulator of vitamin A forming enzyme BCO1 and ISX target genes carbon. We found there is a complete downregulation of vitamin A forming enzyme BCO1 and this carbon in the vitamin A sufficient and the beta carotene compared to the vitamin A deficient diet. So this data clearly shows that, due to the downregulation of beta carotene oxygenase 1, there's an intact vitamin A, intact beta carotene observed in the intestine were unable to convert it into vitamin A. Next, we would like to measure the vitamin A levels in the other part of the tissue of the LRAT knockout mice. The reason is this LRAT knockout mice, due to the knockout of LRAT enzyme, it is unable to store vitamin A in the liver. But this mice can able to store high level of vitamin A in the fat, that is like white adipose tissue. The reason is the enzyme DGAT. So it's a DGAT dependent. In the DGAT was highly active in the fat. As we measured the vitamin A level in the fat, we found there is a high level of vitamin A in the vitamin sufficient diet, but a very low amount of vitamin A in the beta carotene supplemental LRAT knockout mice, and there is no vitamin A in the vitamin deficient mice. So this data alone clearly shows that LRAT knockout mice has a problem in the beta carotene metabolism. Beta carotene metabolism in the LRAT knockout mice. This data was similar in the liver. It was similar in the levels of vitamin A in the liver retinol, but interestingly, we found a very high accumulation of liver beta carotene. This data clearly shows that due to the dysregulation of ISX, this LRAT knockout mice were unable to convert intact beta carotene into the vitamin A, because this dysregulation of ISX leads to the complete downregulation of vitamin A forming enzyme BCO1. This data clearly shows that due to the dysregulation of ISX, this beta carotene were unable to converted into vitamin A in the LRAT knockout mice. Next, we would further like to show that ISX was regulated by retinoic acid signaling. To prove this concept, we would like to block retinoic acid signaling and measure the ISX expression. To prove this hypothesis, we would like to utilize retinoic acid receptor antagonist and block the retinoic acid signaling, and we believe that blocking of retinoic acid signaling can downregulate the ISX expression. Thereby, we can able to restore vitamin A forming enzyme BCO1. This is the retinoic acid receptor pan-antagonist we use for our study. For our study, we use LRAT knockout mice fed with beta carotene for 1 week. The reason we use beta carotene diet is when we fed the LRAT knockout mice with beta carotene, there was a upregulation of ISX that leads to complete downregulation of PCO1 vitamin A forming enzyme. So after a week of beta carotene diet intervention, we gavage those mice with retinoic acid receptor antagonist for 2 days at the concentration of 5 milligrams per kilogram. After 2 days, these mice were sacrificed. Previously in our lab, we used the whole intestine for our study, but recently for this study, we would like to go for a detailed examination. For that, we isolate villi and crypts separately and use for the qPCR experiment. After the retinoic acid receptor antagonist intervention, we isolate villi and crypts and use for qPCR expression. After this intervention, this retinoic acid receptor antagonist blocks the retinoid signaling. Thereby, this ISX was completely downregulated in the intervention group compared to the control. So this data clearly shows that this ISX was regulated by retinoic acid signaling. Hence, due to the downregulation of ISX, this ISX target gene SCAR1 and vitamin A forming enzyme BCO1 was recovered, and it got upregulated up to 500-fold in the BCO1 and 15-fold in the SCAR1, both villi and crypts. This data clearly shows that ISX expression was completely regulated by the retinoic acid signaling. Further, we would like to determine this LRAT knockout mice unable to utilize beta carotene as a sole source for vitamin A is due to the ISX dysregulation, and also to measure to make LRAT knockout mice to utilize beta carotene as the sole source for vitamin A. We carry out genetic rescue experiment. For this genetic rescue experiment, we are interested to make a double knockout mice where LRAT and ISX knockout mice were breed together to get LRAT ISX double knockout mice. So for this experiment, this LRAT and LRAT ISX double knockout mice were fed with vitamin A deficient diet for a week. After washout period, these mice were fed with beta carotene as a sole source of vitamin. Next, we measured the different tissue retinoid level. This genetic rescue experiment exponentially increased the vitamin A level in the gut, liver, and especially in the fat. You can see here it has about 10 nanomole per gram of vitamin A in the ISX LRAT double knockout mice compared to the LRAT knockout mice. So our genetic rescue experiment clearly shows that knockout of ISX in the LRAT can able to recover vitamin A levels. And next, we would like to measure the knockout of ISX can able to recover the vitamin A metabolism enzymes in the intestine. So next, we measure the expression level of BCO1 and SCAR1. We found that due to the knockout of ISX, there was about 100-fold increase in the expression of vitamin A forming enzyme BCO1 and ISX target gene SCAR1 in the double knockout mice compared to the LRAT knockout mice. So next, we would like to measure the retinoic acid metabolism gene in the liver. Here also this type 26A1, which are the retinoic acid catabolism gene that is about 1,000-fold increase in the double knockout mice compared to the LRAT knockout mice fed with beta carotene, and this retinoic acid receptor is about 4-fold increase in the double knockout mice compared to the LRAT knockout mice. This data clearly shows that genetic depletion of ISX signaling restores vitamin A metabolism gene in the double knockout mice. Now we come into the final stage of this talk. The summary of the talk is LRAT mice is unable to utilize beta carotene due to the dysregulation of ISX, and the pharmacological intervention of retinoic acid receptor pan antagonist can able to block retinoid signaling. Thereby, it suppresses the ISX expression, and that leads to restoration of vitamin A forming enzyme. Next, the genetic rescue experiment clearly shows that knocking out of ISX restores BCO1 expression in the LRAC knockout mice with beta carotene. And this LRAC is critical for the control for retinoic acid homeostasis in the intestine. And the take-home message is LRAC play a critical role in intestinal retinoic acid homeostasis, and it protects the immature shutdown of intestinal provitamin A metabolism mediated by this ISX dysregulation. By the pictorial representation in the normal physiological process, when beta carotene were absorbed by the transmembrane protein SRB1, and this one molecule of beta carotene were converted into two molecules of retinaldehyde, and most of the retinaldehyde were converted into retinal esters by the enzyme LRAT, minimal by the DGAT1 enzyme. And these retinal esters were further packed into chylomicron, and it was distributed for liver storage and normal distribution. And in other side, there's retinoic acid. This is important for normal expression of more than 500 genes. But in the absence of LRAT, this step was completely seized out. So all the incoming beta carotene were converted in retinaldehyde, and further it was more oxidized to retinoic acid. Increased retinoic acid concentration prematurely increased the upregulation of ISX. That increase in the ISX upregulation that leads to complete downregulation of vitamin A forming enzyme BCO1. Thereby, beta carotene were absorbed by the another way by CD36, and it accumulates in the intestine, unable to convert into vitamin. But with this, I would like to acknowledge our lab members, especially our lab head Johannes von Lintig, and lab members, past lab members, and our collaborator Dr. Marcin Golchak, who is the discoverer of the structure of the LRAT knockout, that's the LRAT enzyme, and also our funding National Eye Institute. With this, I am ready to take any questions. Thank you.
Thank you, sir, for your wonderful presentation. Sir, we have certain question posted in the comment
Okay.
section.
Okay, I will check. Okay, yeah.
Yeah, sir. Yeah. Sir, as this carotenoids, it's fat soluble.
Yeah.
Does it have any impact in the liver absorption?
Yeah, actually, beta carotene can able to accumulate in the liver, and high accumulation of this beta carotene can lead to liver cirrhosis. And if knocked out this beta carotene oxygenase 1 enzyme, in over time, there was this mice, the BCO1 knockout mice, can lead to liver cirrhosis. In our lab, we have the data which shows that beta carotene oxygenase 1 mice, which can able to accumulate high level of beta carotene in the liver. In older age, these mice have a lot of problems in their lipid path.
Yeah, that's fine, sir. Whether this LFAT enzyme is correlated with this liver absorption, and one more query from the participant, that's how about the carotenoids, beta carotene from Dunaliella selenites and green algae have impact on the therapeutic value?
I'm really sorry for that. Actually, we are not working completely on the beta carotene. We are working on more on the vitamin A. So we are not working on the intact beta carotene. I'm really sorry. I don't have answer for this question. Sorry.
That's fine.
Yeah.
So actually, we have one more question from the participant.
Yeah.
Is the ISX antagonistic can be used to treat beta carotene deficiency?
This beta carotene deficiency.
Yes.
I don't think so. Beta carotene, it cannot recover beta carotene deficiency because this retinoic acid does not have a direct correlation with the beta carotene oxygenase 1 enzyme, because retinoic acid is regulated to the ISX. If you block a retinoic acid, that blocks the ISX expression, right? So that means it upregulates the BCO1. So there's all the beta carotene when we are getting into the intestine were converted into vitamin A. So beta carotene will not accumulated in the intestine. So it is opposite actually.
Thank you, sir. Hope you have clarified all the queries. And it's indeed an excellent presentation, and we are very happy on your extensive informative data which you have worked on beta carotene. Thank you, sir.
Thank you, ma'am.
So, dear participants, we are in the final part of the two-day international webinar on current scenario in therapeutics, prospects and challenges. With this, I invite Dr. P. Ramesh, Head Department of Botany, to brief our seminar and to deliver the vote of thanks.
A very pleasant good evening to all. I feel immense pleasure of gratitude to lead the seminar report of the students' international webinar on current scenario on therapeutics, prospects and challenges. Around 1,400 participants from various colleges and universities participated in the webinar. The day one of the webinar started on 18th March 2021 with the inauguration at 2:50 p.m. Dr. G. Jodi, Dean Life Sciences, have the brief note about the college, followed by welcome address Dr. T. Kavita, Head Department of Microbiology, Dr. S. Srividya, Director.
Sorry for the interruption, sir. You do continue. Hello, sir. Hello. Sir, you 3 minutes to flow it, sir. To flow come time 2 seconds, sir. Please hit, sir. Okay. Thank you, sir. Sir, okay. First week was not working, sir. Network, sir. Network. On the power voltage. On the power voltage. And the power voltage.
UPS
இல்ல இது தனி பவர் கொடுத்துறாங்க கட் பண்ணாம அது தனியா பவர் கொடுத்துறாங்க அவங்க சொல்லணும் டேவிட் சார் டேவிட் சார்
இது போடணுமா?
பவர் ஆன் லைன் வந்தால் இருக்க சூழ்நிலை வெளில போயிட்டா நெட் கட் ஆயிடும், ஸ்டீம் கட் ஆயிடும், லைவ் ஸ்ட்ரீம் கட் ஆயிடும். வெளில போயிட்டா
இப்ப என்ன சொன்னாரு?
அது போகும்
நான் இருக்கிறேன்.
சார் அது போகும். அப்படின்னு வெளில போயிட்டா மீட்டிங் கட் ஆயிடும். நான் வெளில விட்டுருவேன். அவர் தான் போயிடும். அவர் சொல்றேன்.
இது என்ன பண்ணணும்? நான் டேவிட் இமீடியட்டா பவர் போடணுமா?
எல்லாரும் பவர் பட்டன் வருங்க.
இமீடியட்டா இது போட்டுடுங்க சார். கரண்ட் வந்துச்சா? உங்கள கரண்ட் வந்துச்சு.
கவிதா மேடத்தோட நிற்குது. கவிதா மேடத்துல வந்து படிக்கணும். கவிதா மேடம் முடிச்சிட்டு வெல்கம் ஸ்டார்ஸ் அதுல இருந்து அப்படியே பண்ணணும்.
Dr. S. Srividya, Director, Academics, inaugurated the webinar with her words of wisdom. Dr. J. Radhika, our beloved principal, felicitated the webinar with her generous words of the blessing and appreciation. The keynote address started at 2:30 p.m. Dr. Sasikumar Ponnusamy, Postdoctoral Associate, University of Buffalo, New York, USA, have delivered a lecture on natural products and effective approach to fish parasites in aquaculture. His session ends with interaction from the audience. The second keynote address started at 4 p.m. Dr. S. Kokul Shankar, Senior Associate Professor, Microbiology Unit, Deputy Dean, students and alumni, Faculty of Medicine, AIMS University, Kadak, Malaysia, have delivered a lecture on fungal infections, overview of current scenario, followed by his keynote address. Dr. P. Thirumalaivasan, Head, Department of Biotechnology, have the vote of thanks to such a distinguished speaker, intellectually stimulating the audience. The day second of the international webinar started on 19th March 2021 at 2:30 p.m. Dr. Sasikumar Ponnusamy, Postdoctoral Associate, Department of Oral Biology, School of Dental Medicine, University of Buffalo, New York, USA, have delivered the keynote address on molecular renovation for engineering probiotics for the human therapeutics. The second innovative lecture was started at 4 p.m. Dr. Research Associate, Department of Pharmacology, Cass Western Reserve University, OKU, USA, have delivered the keynote address on regulation of vitamin A homeostasis of the intestinal barrier. The session ends with the active interaction from the participants. Dr. T. Ramesh, Head Department of Botany, have presented the seminar report of the 2 days international webinar. The webinar ends with the vote of thanks. Thank you all.
Thank you, sir. Dear participants, the feedback link has been posted in the chat box. Please go with the link. Now it's time to deliver the vote of thanks to the audience.
Good evening all. I feel honored and privileged to get the opportunity to propose a vote of thanks on this special day. We have hosted the 2 days international webinar on current scenario in therapeutics, prospects and challenges with 4 eminent resource persons and around 1,400 participants from various colleges and universities across the world in all streams of education from science to technology and research. On behalf of Srimad Andavan Arts and Science College, Trichy, Tamil Nadu, India, I thank all the distinguished delegates who have accepted our invitation and spared their generous time with us. Thank you for your very interesting and informative lecture. Thank you all. I thank today's speaker, Dr. Sasikumar Ponnusamy, Postdoctoral Associate, University of Buffalo, New York, USA, and Dr. Ram Kumar Srinivasagan, Research Associate, Case Western University, Ohio, USA, for their valuable inputs. Also, I extend my sincere thanks to Dr. P. A. Venmadi Maran, Associate Professor, University of Malaysia, Shaka University, and Dr. S. Kogul Shankar, Senior Associate Professor, Faculty of Medicine, AIMS University, Malaysia, for sharing their expertise. I would like to propose hearty thanks to our Honorable Secretary and Correspondent, Amangi Balaji, for his wishes and support towards the successful completion of this webinar. I thank our respected Director, Dr. S. Srividya, for gracing the international webinar with the inaugural address. Thank you for your sharing with us your words of wisdom. We have been fortunate to be guided, our principal, Dr. J. Radhika, for your continuous support, and you have given us a lot of encouragement. I would like to thank Dr. M. P. Chaimani, Vice Principal and Dean of Commerce and Management, who have blessed us with his presence. I extend my gratitude to the organizing team, MC members, staff members of Life Sciences, non-teaching staff, technical team, automation unit, and supporting staff for the successful completion of this webinar. I like to express my sincere thanks to Get and his team of Visual Communication Department for the splendid support of this 2 days event in effective live streaming. Last but not least, I thank all the delegates, students, and participants for the irresistible, responsible, and the interest towards this 2 days international webinar. I once again thank you, everyone making this program a grand success. I would like to thank you all very much. Jai Hind.
Thank you, sir. Dear participants, you will get the certificate to your registered mail ID probably in about 2 days. So, hope you all have enjoyed the sessions, and we hope the sessions we have lined up is fruitful and informative. With this, we thank one and all, and extend our warm gratitude to the convener and everyone for organizing such a wonderful webinar, and special thanks to the active participations of the participants and also the speaker. Thank you.